t-shirt. It was so weird.
Yeah.
He like walked down the building and he was like baggy shorts. I was like,
This is wrong. All right, let's
it
see.
is.
Oh, I need eye clicker too.
Oh.
So what else do I need?
I have a bunch of questions to ask you about the chapter 9 stuff, but
Okay.
I'm gonna ask you in lab or after lab or something.
Yeah.
Because it's too late. I wanted to come early, but I forgot my water in my car and I had to go back.
That's okay. I usually don't have time like right before class.
I know.
So, especially not on Fridays.
It's just Friday.
Fridays are.
It's not really a bunch of questions. It's just like some little questions. But anyway.
And if you want to email me, if you want to.
Yeah, I was
I
gonna
think
email
we'll
you.
probably- we might have time in lab. We might not though.
Okay, well,
We'll see.
if we do. I took
we do-
screenshots of them because I didn't want to forget which slides and all, so
Yeah.
I'm prepared.
Awesome. Okay, so don't forget to go ahead and sign in. This one sock just keeps falling down, and I don't understand.
You should wear long socks.
I hate long socks.
I only wear long socks.
All right, so don't forget, do your attendance.
But it's definitely your separation.
Five,
So one of those is the,
5 pairs.
there's this product.
And the rest
Or long.
and
And fun. I do like fun socks. I like fun socks.
That was the.
I have a weakness for fun socks. Okay,
Oh, yeah.
let me scroll to where we are not looking at the answers. All right.
I don't know if we did that much separation.
Full screen.
It's like stone.
Okay, make sure you have a sheet of questions.
The last one, like this is
Was it?
when I was studying, I was remembering,
Good.
like, when you were like, why is I still have all this?
I wasn't just
But
kidding.
it did help me remember the 3 things, like,
Yeah. You're like, what was she talking about?
like when you were saying it, I was like, but then when I was studying, I was like,
So the rate determinants we got for glycols are governed by.
Delta G. Why? Because they're not governed by substrate concentration.
Those are the rate determinants.
So we're going to talk about it again when we talk about citrate. It'll reinforce. Just stick with it. All right. So what's a pirate's favorite letter?
R.
You'd think, but he couldn't get past the C.
that was actually good.
He had to have 2 today. He wanted 2 today. Do you want the other one?
Yeah.
All right. What do clouds wear?
Clouds?
Clouds.
Like in the sky.
Yeah. Thunderwear.
That sounds like
Classics today. Classics. All right. So I just handed you a couple of questions on chapter 10. I didn't really hand it to you, but you picked one up. So I want you to try not to look at your notes. These questions are all about basically the conversion from glycolysis to citrate. So things that happen in between. So try to answer them. Then we're going to answer them together. You can work together. You can talk to people. Either way is fine with me. Grab a sheet. Grab a sheet.
Thank you.
Yep. So read the questions and try. I'm going to be quiet. It's very difficult for me. Oh, and B should be 3. I'm sorry. What are the 3 fates of acetyl-CoA? I forgot I made that mistake. 3. That help? I'm sorry. I'm sorry. Do you still need the recording?
Just like the last 30 minutes. 20 minutes.
Send me an email and I'll reply back and send it to you. Same thing. Oh, did I post it? I wanted to. Let me look. While y'all do that, keep going. Keep going. Don't stop. I meant to make one. What's that?
Sorry.
Trying to keep y'all rhythm going. Let's see, did I? No, I didn't. I still need to do it.
I've been wondering about
I still need to do it. Okay. Oh my gosh, I said I'd do it and I didn't do it. I'm gonna make a grade calculator specifically for this class so that y'all can put in y'all exam scores and calculate what you'll get for a final.
Are we gonna know like the grade for this exam before we take our final? Because that's gonna like play a factor.
That's gonna play, and I'm gonna get them to you as soon as humanly possible.
Okay.
As soon as humanly possible. I appreciate the
I
offer.
think Zach has some free time.
Yeah, because y'all have so much free time, right?
Yeah. A lot of it.
All right, let's try this. Y'all are so quiet. Do y'all want to talk to each other before we try this? No one wants to try by themselves. Okay, so let's talk about A. What drives redox reactions? Reduction potential. Okay, so that's a general concept. That doesn't really answer the question, right? That's a concept. If you put that on my exam, I might give you a little partial credit, but you really have to explain it. So how does it work? Right? That's the question. What's that? Yes, yes. So the reduction potential of individual molecules determines if it's an acceptor or a donor. So let's make a sentence about donors and recipients in terms of reduction potential, right? What's the relationship between the two? An electron donor must have I'm gonna put a blank, reduction potential than the recipient. Recipient, whatever. Y'all know what I mean. So how do the electrons flow? From a low to a high. Okay, so the electron donor must have a lower electron reduction potential than the recipient, right? Okay, so let's think about this. This is a good statement. We're on the right track. Is one reaction good enough in order to harness the energy that's stored in those molecules? Like, can I transfer electrons once and like, woohoo, I made ATP, I'm good, right? No? No? No, absolutely not, right? Okay, we got to go through a lot of different reactions. So we're going to make NADH, we're going to make FADH2. then where are they going? They're going to the mitochondria. They're going to enter into the ECT, the electron transport chain, right? Now, they don't just enter in and then we make ATP out of that, right? It's sequential reactions. So anytime that you're trying to convert energy from one form to another, one reaction is not enough. You're going to have a stepwise series of reactions. So you must have a stepwise series of what kind of reactions? Redox. Right? And so that we're incrementally increasing that reduction potential. So we incrementally increase the reduction potential. What's our final electron acceptor? O2. O2 has the most positive reduction potential. It's not very high, plus 0.82. It's not high, but it's high enough. It's higher than all the other ones. That's why oxygen is the final electron acceptor, because it has the highest reduction potential. I'm trying to get y'all to say the words. Okay, so does this make sense? Yeah, so what can you expect when we get to the electron transport chain? That those electrons that are in NAD, NADH and FADH2 are going to be transitioned multiple times. We're going to see many, it's like passing the hot potato, right? Passing it along, passing those electrons along. Okay, what was our next one? What are the 3 fates of acetyl-CoA in the cell? Okay. So in order to remind us of this, if I have glucose, glucose goes through glycolysis. What do I make? I make pyruvate. Can pyruvate enter the citrate cycle? No, what are we going to do to it? Acetyl-CoA. Now that we have acetyl-CoA, what can we do? Go into, we'll just do this, right? We'll put it citrate. So what did you just tell me? You just told me one of the fates of acetyl-CoA. What's one of the fates of acetyl-CoA? So enter citrate. And then eventually you'll go to electron transport chain. Then you'll go through ATP synthase, right? And eventually you want to make ATP. Right? So it's kind of like the end fate, if you want to think about it that way. Okay, so what else? What if I go on a low carb diet? I should, but I like my bread way too much. My triglycerides will be converted to what? Oh, so what if I eat too much carbs? What's acetyl-CoA going to do? Going to make fatty acids, triglycerides, triacylglycerides. So what's fate number 2? So number 2, I guess I should number that. Number 1, y'all knew this stuff. Y'all know y'all knew this stuff. You just sat there and looked at the words and it didn't come to you, right? So when would we make fats? Tell me about the cell. Don't tell me about what I just had for dinner, but tell me about the cell. I have a high energy charge. A high energy charge. right? That's AKA I've eaten too much carbs. High energy charge, acetyl-CoA, shunted to make fatty acids or triacylglyceride, right? Okay, so that's 2 amino acids. Okay, now amino acids. Is this the only thing that happens? I'll just abbreviate that AA. Is it like that? No, it's a little bit different. Why is it a little bit different? What else can we make amino acids out of? Pyruvate. Pyruvate. So, well, it would actually be, I'm trying to erase. So it actually looks like this. I could do this, right? Right? And then like back and forth that way. Okay, so number 3 is under what conditions? Well, let's think about this. Let's think about this. When do we want to make amino acids? We don't have any. When we don't have any. So when we have low amino acids, that's definitely one. What are other cellular conditions that are required for us to produce amino acids? Low energy charge or high energy charge? High. Fats are kind of like you have a high energy charge and you're not low on anything else. So fats are like the final pathway that you're going to take. But if you need anything else, you're going to shunt it off to go do that first. Make sense? Yeah. Okay. So that's our 3. What else we got to answer? Is this helping? Okay. Why is it so? Oh, I hate when it does that. Okay. All right. What are the required components of pyruvate dehydrogenase? I want to know subunits and I want to know cofactors. Okay. Scroll down. All right. So what do we got? Subunits. Pyruvate dehydrogenase. These are really easy. You number 1, 2, 3, and pyruvate dehydrogenase is an enzyme, so it's yay, right? Do you remember that? It was the circles, right? It was 3 kind of big circles and then one kind of down here, and everything kind of went in and out. You remember that? I have to go back and look at that. It's in the animation. It's in the animation. Yeah? Yeah, okay. All right. So then what else do I have to give? Cofactors? Right? I said give, what did I say in the question? Give subunits and cofactors? Yeah, okay. So I have 3 subunits. Now I have cofactors. Cofactors are things that are required for pyruvate dehydrogenase to function. So a lot of times, what do we call these? Vitamins, right? These are vitamins. They just have scientific names in addition to the vitamin common term that we use. So what do we got? Somebody said NAD. I heard NAD. Yeah. Yeah. And that's B3. You don't have to know which B vitamins are which, but it's kind of interesting. Okay, what else? FAD. FADH2 is the no. Okay, so FAD is B2, then what? you're making acetyl-CoA, so you need CoA. So CoA is B5. What else? Then it gets kind of tricky. The last two, like, you kind of have to memorize. Sorry. The other, these make sense, right? Okay. They're like, yeah, sure. T-H-I-M-I-N-I-A-M-I-N-E? Yeah. Pyrophosphate. Pyrophosphate. This is B1. So when I'm writing the in parentheses, if you consume these vitamins, your body will process these vitamins into these cofactors. It's not like they're the same thing, but if you consume the vitamin, then your body processes it into the cofactor. Okay, and then what's the last one? Alpha OAC, lipoic? No. IC. LIP, right? LIPOIC. IC. And this one is not a vitamin because we make it. We make small amounts of it, but we make it. Okay, so what, did I ask this? No, I didn't. But what makes a vitamin a vitamin? Like this is a legitimate biochemistry question. What makes a vitamin a vitamin? How do we know that our body needs it? He's right. It's something that we need, but our bodies don't produce it. How do we know that we need it? It's an interesting question, right? How do we know that we need it? People who are missing these things in their diet will have medical conditions that can then be treated by supplementing with whatever vitamin they're missing. Yeah? Okay, so that's how we define it. We define it as something that if it is missing, it causes a medical condition, and we can treat the medical condition just by giving you that vitamin back, right? So why don't we make it? If we need it, why don't we make it? We can get it from external sources. So typically vitamins are huge. Vitamins are energetically a lot to make. So if we can eat it, then we don't have to make it. Great, right? Awesome. So who makes it? Plants, bacteria. Most other animals are dependent upon it in their diet. So it's an evolutionary thing, right? Well, if plants have to make it because their food source is light, so they have to make everything else. They have to make everything else. If we eat the plants, then we don't have to make it. It's kind of cool. Okay. That was my side trip for the day. I do that often. Okay, how is pyruvate dehydrogenase regulated in the cell? I have to go and remind myself of what I'm asking. Okay, so how do we regulate it? There are two ways that we can regulate it. Okay, so say it again. Covalent. What's the most common covalent modification? Phosphorylation. That's the only thing you're going to remember from this. And phosphorylation actually is a negative regulator, an EG. Negative regulator. Okay. So if we phosphorylate pyruvate dehydrogenase, it won't work as well. What else? Feedback inhibition, which is a type of allosteric regulation. Good. Okay, so what will feedback inhibit pyruvate dehydrogenase? What's the product? Let's wait, let's write the reaction. Okay, I guess I'll write it here. So what do we need? Pyruvate plus, what's the product? Acetyl CoA, so you need CoA. All right, now what? We need to put in NAD. NAD. It's just my brain goes faster than my pen can write. That's my story, and I'm sticking to it. What else? That's it. Now what are we gonna make? Acetyl CoA. We're gonna make NADH. I'm running out of room. Hydrogen ion. When we make NADH, we'll have a hydrogen ion. What else? How many carbons get transferred from pyruvate? How many carbons are in pyruvate? 3. How many carbons are transferred in that first reaction in the citrate cycle? 2. Carbon dioxide. Yeah? Okay. So allosteric regulation is a way that we can regulate this reaction. So what do you think is going to feedback inhibit pyruvate dehydrogenase? So negative regulators from allosteric. What's a product? Acetyl CoA. Right? Think about your products. What else? What's another product? Oh, y'all are onto something now. Okay. And what else? I keep hearing it. I thought you said it. ATP. Why do we want ATP? ATP is not, it's not feedback. These two are feedback. But ATP is still a negative regulator of pyruvate dehydrogenase. And that almost doesn't make sense. Like, why would energy be a negative regulator of an enzyme? But this is an enzyme that's in an energy making pathway. So if we have lots of energy, a high energy charge, we want to stop making acetyl CoA, right? Good. Okay. Okay, so why do we want ATP to be a negative regulator of pyruvate dehydrogenase? Doesn't quite make sense, right? Because normally most enzymes require energy to go. But in this case, because pyruvate dehydrogenase is in the pathway to generate ATP, technically it is a feedback. It's just way downstream, right? So it's saying that our energy charge is really high, so you don't have to go through citrate anymore, right? You could do other things. Yeah, cool. Let's go talk about the reactions in the citrate cycle. Where was I? Here, right? About?
I think we stopped on the very first.
The very first one. Okay, so remember, if you have a very high negative delta G value, what does that tell you? That you are irreversible. And how are you regulated if you are irreversible? Are you regulated by substrate concentration? No, no, indeed not. You are regulated by covalent modifications and allosteric regulation, so binding of other molecules. Okay, so it's important to know which ones have high negative delta G values. It's important to know which ones are generating our energy carrying molecules. What are our energy carrying molecules? NADH and FADH2. It's important to know which ones are generating energy in the form of GTP, which is an ATP equivalent, right? Okay, so let's go through and talk about some of these individually. So when we look at the whole cycle, right? We're going to start with pyruvate. Remember how I said pyruvate is 3 carbons? We let go of one of those carbons when we convert it to acetyl-CoA. Now we have 2 carbons. Those 2 carbons are then attached to what molecule? Come back here. So to do the first reaction to make citrate, we make citrate. Citrate is the product. We have to start with the end product of the citrate cycle. So the last thing that we make in the citrate cycle is oxaloacetate. So in that very first reaction, we take the product of the citrate cycle and we add acetyl-CoA to it. That's what makes it cyclic. Okay, so I'm going to hook these 2 carbons from acetyl-CoA onto oxaloacetate and make citrate. And then we're going to follow these carbons all the way around. So let's kind of look just generally before we go through each individual step, right? So in reaction 1, we're adding 2 carbons. We make citrate. Did we remove a carbon in the second reaction with a conitase? No. What happened? Just kind of rearranged it, right? So it's an isomerization reaction. Okay. So now we have isocitrate. Look what I released. I released carbon dioxide. So I actually let go of a carbon. And when I let go of a carbon, what did I make? NADH. When I let go of a carbon, I released NADH. Oh, okay. All right. All right. Now I'm going to go from isocitrate. That takes me from isocitrate to alphaketoglutarate, right? I lost a carbon. When I go from alphaketoglutarate to succinyl-CoA, what happened? I released a carbon and NADH. Oh. Then I go from succinyl-CoA to succinate. What did I lose? GTP. The important thing there is the GTP. Then I go from succinate to fumarate. This is how I remember this one. When you make fumarate, what does fumarate start with? F, what does it release? FADH2. That's how I remember which reaction releases the FADH2 versus an NADH. Okay, so you know it's fumarate. So then when we go from fumarate to malate, what do? Hydration, right? Yes, with me? Then when we go from malate to oxaloacetate, what do we release? One more NADH. Now this one does not release a carbon, okay? It's the only one that releases an NADH and does not release a carbon. Only one. And now I have my substrate that I'm going to use in my first reaction. So when you look at this, when you look at our insanely high delta G value here, you would think that this reaction wouldn't go, right? This is the malate dehydrogenase. So here's malate dehydrogenase. Why does this reaction proceed with such a high delta G value, such a positive delta G value? I'm using up the product in the next reaction. So how much oxaloacetate is really there? Not much. It doesn't last for long. We're going to use it up real quick, right? Okay. All right. We did the animation, but you should go back and do the animation again once we go through the whole thing. All right. So reaction 1, citrate synthase. Tell me about citrate synthase. Very high negative delta G. Delta G 31.4. So what do I know about this reaction? Irreversible. Should be able to pick that out. So this very first reaction, what we want to do is we want to commit our carbon units of our acetyl-CoA to the citrate cycle. So this is the commitment phase. This is the one that's going to say, okay, now that we've got all these carbons, we can go through a process called oxidative decarboxylation. So what is oxidative decarboxylation? We're going to go through redox reactions and then remove carbons. right? Oxidative decarboxylation. If you just kind of listen to the words, you know what it is. So this is irreversible, so it's very, very highly favorable, right? Yes? Sometimes y'all just look back at me and I have no idea if y'all are with me or not. All right, so if this is highly favorable, okay?
So irreversible. highly
Favorable. Mhm. So if it's irreversible, it's highly favorable. We're not going to regulate this with substrate concentrations. So tell me, what would be a positive regulator for this reaction? What do you think? If high energy charge is typically a negative effector, then the opposite must be true, right? Low energy charge must promote it. So how do we know we have low energy charge? ADP, right? The opposite of ATP. Some of them will be ADP, some of them will be AMP, but either one is high when ATP is low. So then how about negative regulators? What do we think? Opposite would be ATP, right? That makes sense. What else? How about the product? Feedback inhibition, right? If citrate builds up, that means my other reactions are backing up. So my electron transport chain must be overloaded and overworked, right? So I don't need to make any more. What else do we make in the citrate cycle in general? Like what's the goal of the citrate cycle? This is a really good question. What is the overall goal of the citrate cycle? To make energy carrying molecules. Energy carrying molecules are NADH and FADH2. In this case, NADH will inhibit it. And then succinyl-CoA will as well, but we haven't really talked about that yet. So you could put it, C-I-N. We haven't talked about that yet, but we will. Okay, so it's our very first reaction. Succinyl-CoA. Succinyl-CoA, sorry. I'll just tell me. All right, reaction 2, aconitase. What do you see with these arrows for aconitase? This is what kind of a reaction? This is a reversible reaction, very reversible. So it depends on what? Substrate concentrations. Y'all are starting to get the hang of this, right? So this is really just an isomerization, and you have an intermediate, but that intermediate doesn't hang out for very long. So we don't normally show it when we show it in the citrate cycle. We just show citrate isomerizing into isocitrate, right? Okay, so all you do to form it is we go through a dehydration event and then we go through a hydration event. And what happened to our carbons? So the first carbon, we remove this hydroxyl, the first carbon, I say really the third carbon, right? The first carbon in the box. We lose the hydroxyl, so we get a double bond between the two carbons. Then when we rehydrate, that hydroxyl moves to the next carbon. So it's just an isomerization reaction, okay? All right, now reaction 3, isocitrate dehydrogenase. What do we know about isocitrate dehydrogenase? Dehydrogenase, when you hear that for an enzyme, what should you be thinking? Oxidation reduction, right? And so we're going to be producing some important molecules here, right? So they probably, this reaction probably has a very high negative delta G. We want to do that. Delta G negative 20.9. You don't have to memorize the delta G values. You just have to know which ones have a very high negative delta G. So this would be an example of a irreversible reaction. So it's favorable. We said that before, right? It's favorable. So this is our first example in the citrate cycle of an oxidative decarboxylation. So why do we call it an oxidative decarboxylation? Well, what happens in this first, if I scroll down, in this first part, right? We're going to make an intermediate here, but what do? What kind of a reaction is this? Oxidize alcohol to a ketone. And so, and in the process, we convert NAD plus to NADH. So this is an oxidation reduction reaction, right? Okay, so then what happens in our second reaction? Decarboxylation, right? So this is an oxidative decarboxylation reaction. That's why they call it that, right? Okay. Let's see. The other thing that's kind of important to know is you can actually go through part of the citrate cycle and make alphaketoglutarate. Alphaketoglutarate is really important if you take biochem 2 in a lot of other pathways. So alphaketoglutarate is a substrate in other pathways. So this is another point at which you can shunt things off. You can go and make other things. Okay, so if this is a highly favorable irreversible reaction, what is this enzyme going to be inhibited by? So minus ATP, NADH, okay, and not so much the product in this case because it can be shunted off to other pathways. We don't want the product to inhibit the enzyme in this case because it can do that. So how about positive effectors? Opposite of ATP, ADP. Now, there's another interesting positive effector for this enzyme, and that other one is calcium. So why do you think calcium, I'm writing the 2. See, my brain just goes too fast. That's all it is. So why would I want calcium to be a positive effector of an enzyme in the citrate cycle? What happens when I want my muscles to contract? Do you remember we talked about this? You have calcium, you have massive amounts of calcium that are going to flow into the cell, right? So does it make sense for calcium to be a positive effector of this cycle? It's indicating time when muscles are contracting and require high energy levels, right? So it's a signal of what the body's doing. Okay. All right. So next, alpha-ketoglutarate dehydrogenase. A dehydrogenase is in redox reaction, and so we have another oxidative decarboxylation reaction. So remember how we had in pyruvate dehydrogenase, we had E1, E2, E3. It's the same basic setup. It's the same type of enzyme. So it has those same cofactors and the same mechanism. And so we eventually convert alpha-ketoglutarate into succinyl-CoA. So what do we think about this reaction? Even if you didn't remember, what would you say for a delta G value? Very negative. Very negative. Negative 33. So that means it is highly favorable. My gosh, you're seeing the same thing over and over again, right? Okay, so if it's irreversible, highly favorable, is it going to be controlled by substrate concentration? No, so we're going to have some positive and negative regulators. Give me some negative regulators. NADH, ATP, also succinyl-CoA, which we'll talk about. It's S, it's an S, it's just an S. Sorry, terrible handwriting on this. That insinuates that I have good handwriting otherwise. All right, so how about positive effectors? What? No, not CoA, not CoA. You would think, but it's not CoA in this case. So it's not ADP, in this case it is AMP. It is AMP, but I would, you can just say low energy charge, and that covers both AMP and ADP. So like, I'm not gonna get super nitpicky about that, I'm not. If it's AMP and you put ADP, I'm cool. Okay, I'm not that picky, as long as you understand that that's low energy charge. Yeah, okay. And then what else would be a positive effector that we just talked about? My muscles are contracting, I'm gonna make lots of calcium. Good. All right.
I don't know when to use the like in product as the
You
as on the last one we were just like we don't do it.
you don't do it because of what those substrates can then be used for. So if there are other places that it can be shunted off to, then you can, right? But this one isn't shunted off of anywhere. This, you make succinyl CoA, so you're inhibited by succinyl CoA. So alphaketoglutarate, if you look on this one, remember I said it was a substrate for other pathways. So if this building up inhibited this enzyme, you would inhibit multiple other pathways. Here, we're not using it in any other pathways, so we can have it as a negative regulator. Okay. No?
Not many words, just
It's a lot.
Yeah. Well, can you see this is why I didn't want to start halfway last time? All right, succinyl CoA synthase, synthetase, synthetase, right? Okay, so in this one, right, what are we going to do? We're going to generate GTP, right? That's our main goal. So we actually make an intermediate. So you're actually transferring electrons and you're trying to phosphorylate something, right? You have this inorganic phosphate that we'd like to tag on to GTP. So basically what we do is we put it onto our succinyl CoA and then we transfer it from there onto GTP. So tell me about the energy required to make a nucleotide triphosphate. Doesn't matter which one, doesn't matter if it's ATP, GTP. Is that an input of energy or are you getting energy out? That's an input of energy, right? A lot of times we couple reactions, right, to make it energetically favorable. Well, that's exactly what we're doing in this case, but in the opposite direction. So the intermediate that we make in this process is very, very high energy. So you can drive the reaction through that high energy intermediate to make GTP. So you got to think about it, it's the opposite. So what do we call it anytime we make an ATP, a GTP, anything like that? We call that substrate level phosphorylation. That tells me that I'm making a nucleotide triphosphate. Okay. Ready? All right, now we got succinate dehydrogenase. Succinate dehydrogenase, remember we said this is the one reaction where we're making FADH2, but we're not also making an oxidative decarboxylation reaction.
So how it's producing CoA again? Can that CoA then go on and go back?
Into the citrate cycle in general? Yep, absolutely. Absolutely. Your body recycles everything. Literally everything. Okay, so remember we said when we make FADH2, the other product that we make in that reaction also starts with an F, which is fumarate. So when you make fumarate, you also make FADH2. So is this a high negative delta G value? No, so it depends on substrate concentrations, right? But it is a redox reaction. It is redox, but there's no decarboxylation. Okay. All right. And then where does this FADH2 go? Electron transport chain. So what you're going to find out is that the FADH2 and the NADH go to different places in the electron transport chain, which is then going to end up producing different numbers of ATPs. So they work a little bit different. Okay, fumarase. Fumarase catalyzes a hydration reaction, right? So we have fumarate, we attach a hydroxyl on there, we make this intermediate, this very, very short-lived intermediate, and then we plop on another hydrogen and we make malate, right? So this is really kind of interesting because this malate that we make, this enzyme fumarase is isomer specific, so it will actually make L-malate. So it makes the L isomer, right? Remember, that's the chirality, right? Okay. Then we have malate dehydrogenase, right? If you're going to have a dehydrogenase, what are you going to make? An energy carrying molecule, right? Because it's an oxidation reduction. So you're going to make NADH in this one. So you're going to oxidize your hydroxyl group over here, and you're going to remove that hydrogen, and you're going to make oxaloacetate. We made it. We made it. What did we make? Oxaloacetate. Woohoo! Y'all are not as excited as I am. You want to go again? Oh, no. No. All right. So I want to go back real quick. How much time do I have? Not long. Ooh, I went too far. Right? Okay. So here's our overall picture, right? So what's kind of important to know is that these two carbons that come in here on pyruvate, right? They're here on acetyl-CoA. They come in and they attach to the top of citrate. If you track these carbons all the way through, right? They're still inside of oxaloacetate. So this is why citrate has to be a cyclic cycle, because those initial carbons that you put on are not the carbons that come out, right? Here's one of the carbons that comes out as CO2, right? We still have these two carbons, right? Then what happens? Now we still have these two carbons being our carbons on top, right? So then what's the next carbon that comes off? The next carbon that comes off is here at the bottom. That becomes that CO2, right? So then we still have these two, right? We still have these two. 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 So what's going to happen is the next one is going to come in and tack two more carbons on top, right? So you have to go through multiple rounds of the citrate cycle in order to remove the carbons that you put on from acetyl-CoA. Kind of cool, right? I'm like, no, but okay. Can we go now? That's it. All right. So that's really good. So next time we're going to talk about regulation, and then what happens to other citrate cycle intermediates. And I'll do another little, y'all like that little review kind of thing we did at the beginning? That's been helpful? Yeah? Okay. So we'll do that again. And I have a little something for y'all. And I forgot it today. I have to bring it next time. I'm not telling. I'm not telling. It's a surprise. Fridays are bad for me to remember things. So if y'all are in lab, we're going to the classroom because we have to get on the computer. And good luck. I'm sure you're going to do great.
You're going to the classroom.
We're going to the classroom.
Each one.
The one right next door to the lab.